Okay, this is a continuation, I'm sorry, of the earlier prep. There was an issue, and when I tried to edit it, I just ended up deleting deleting a whole chunk. Um, uh, so that was a failure, but we can start at this point. So in this experiment, there were two different amputations. Oops, amputations done. Why is this not? Okay, there we go. And then, the healing of the wound occurred, and then the blastema formed. And so this was a proximal amputation, and this was a distal one. And note the different coloration here. Whenever you're doing experiments like this, you have to have ways of telling which cells come from the donor and which come from the recipient, right? So keep that in mind. We've talked about it multiple times now, and it's another one of those sort of recurring themes, which is just like when you're doing this kind of experiment and you're drawing conclusions, you have to know which cells came from where. Okay, so in this case, after the proximal blastema forms here, it's removed, and then the distal blastema is grafted in its place, right, on the stump. And so what's interesting about this process is that you will get outgrowth, and you will get the development of the correct structures. The limb reforms properly and everything is fine. But when you look at the pigmentation of the cells, the only cells that the cells that actually came from the distal blastema form are the wrist and the hand and finger bones here. So the distal structures. The others come from outgrowth from the stump, which contains the proximal values, and allows for the more proximal to distal generation of all these intervening structures. So this is very similar to this kind of experiment, except that we haven't added a limb here. We have instead put the distal blastema here, but it behaves the same way, with this distal blastema forming these structures, which is what it would have formed in its if it had been just left to develop properly. And then the stump here forms all the rest of the structures. And once again, if you think about this in terms of positional values, then cells at the blastema sort of if this is level 5 expression, if that's what those cells can express, they are not able to divide and form cells that give rise to more of the signal, only to the same or less of the signal. So if you think about this, this isn't quite exactly the right way of describing it, except that if you think about it that way, it's easier to interpret and understand how these processes work, which is why I'm mentioning it a few times. Okay, and so this is the experimental evidence that sort of explains the previous, or agrees with the slide that I talked about last time. Now, one of the types of properties of the cells that differs from proximal to distal is cell adhesion. We already know that there are gradients of signal, right, in the developing limb, in the embryo, and so they're we know, for example, retinoic acid is part of this whole story in the regeneration, and we'll talk about that in a bit. But we also know that there is differential adhesion, and you can show that the adhesion differs by taking a distal and a proximal blastema from an experiment like this. You take those tissues. If you remove them and you don't graft anything on there, the cells can reform it and still regenerate the limb, right? And if you mix those two cells together, cell types together, connect them, then what happens is each of the cell types stays together. So the proximal and the distal ones stick with each other, right? And then there's this process where the more cohesive structure kind of, maybe because the binding is tighter or something, tends to envelop the other. So the distal winds up sort of more on the inside, and this ends up more on the outside. Okay, so there's a certain behavior, and the behavior of the cells is that they congregate together, the same cell type with the same. So and they connect in such a way that seems to indicate that the adhesion of one cell type differs from the adhesion of the other. PROD1 is a cell surface protein that gets anchored in the membrane through a glycolytic, not glycolytic, a glycosylation anchor molecule. So it's anchored in the membrane, so we know it's on the cell surface. And if you treat the cells with an anti-PROD1 antibody, then you get a disruption of the behavior. Okay. And so we can use this result to explain why this result actually happens, right? This tells you what molecule is responsible for it. Antibodies actually, anti-PROD1 antibodies will bind and cover the PROD1 on a cell surface and prevent it from doing whatever it's doing, right? So we're disrupting that protein. And then in this experiment, if you take a distal blastema and you transplant it into underneath a proximal blastema, when outgrowth occurs of the correct limb, the distal blastema participates in formation of the hand, right? But the cells that have been transplanted, they stick together. They stay together because when you put them in here, their adhesion is different, and so they stick with each other and work as, stay as a group. And then as the outgrowth continues, they migrate to the correct position of the structure they're supposed to form, and they go ahead and form that structure. And again, there will be pigmentation differences, that it's not just color coding on the diagram. You know which of these hands came from which original batch of cells based on the pigmentation in the cuticle, the epidermis, the skin of the developing limb. So we know that this kind of behavior is based on adhesion, and we know which molecule is responsible. So when you put all these three different aspects of the experiment together. So it turns out that the NAG, the Newt anterior gradient protein, there are a number of these in a number of different organisms. So these have been identified in multiple organisms. And NAG is essential for limb regeneration. Remember that connection to the nervous system. It's responsible also for the dependence on innervation as part of the regeneration process. And it turns out it is a ligand. So PROD1 is a receptor for NAG. So that's an important process. Now, or property, sorry. This binds to this, right? So, and the effect of the anterior gradient protein is the promotion of cell division. Now, it is not part of the patterning process, but some of the downstream molecules, PROD1 and some other proteins like retinoic acid and so on, do the actual patterning. So NAG is responsible for the cell division that's required to make the cells for the limb, but then the patterning occurs based on other proteins. And we don't really know exactly how the proliferation is generated. It may be via some other. Surely PROD1 isn't the only protein that it interacts with. And we also know in terms of patterning that there is Hox gene expression. We're going to talk about a couple that are the mice proteins. And what's interesting is that there is some suggestion that in the regeneration process, the distal part of the blastema might be specified before and the more proximal parts later, which is weird. It doesn't. It seems counterintuitive to me, and it's in direct contrast to how limb development works. But if you look at their expression patterns and when and where they're expressed, this does suggest that this might be the case. Just something to keep sort of in the back of your mind. Okay, so this is one of those weird kinds of experiments. So if the hand is amputated from the forelimb and then the stump, instead of just letting it regenerate, if you were to allow it to, you make a very little wound in the sort of stomach area and you connect that limb in there, and then vascular connections, so the blood supply are established, so that tissue is going to survive, and then you make a second incision, and that allows you have the two different stumps. And this is I know it looks sort of gross, but this is the classic experiment that showed that just no matter what the orientation of the tissue is in on the stump, always the distal most structures are going to be generated. So in the case of the anterior part here, the real correct limb, you're going to get the proper limb regenerated because where the cut took place, all the more distal structures are developed, right? And if you're thinking of proximal to distal gradient, then that sort of explains it. But in this case, the proximal to distal gradient goes in the other direction. And so once again, you have a cut, and the cut is at a higher region. And it's almost as if, think about the cells at the cut site here, they don't quote unquote know that there already is these distal structures here, so they go ahead and they just reform everything that is missing, as if the cut happened. This and these two cut sites, they are equivalent in terms of positional information, so the parts that they regenerate are identical. And again, this is this idea that whatever level of expression this proximal part has, the cells that divide and are generated from it can only express the lower values, the more distal values, right? So it doesn't matter that the tissue is inverted, it's turned around. It has no way of knowing that it's turned around, and it just goes ahead and properly generates what should be there. Without this conflicting information. And so again, I find these, like a diagram like this is how I make sure I understand how things work. So basically what you have is the forearm, the real forearm, the stump, looks like this, and basically you're going to regenerate all the lower values. And when you have I'm trying to think of how to, so this is a bit misleading, but suppose you've turned this around, and now you have sort of the same positional value that's sticking out from the lower value. It's also going to generate those lower values. So each of the two stumps are starting at the same point, because the cut site is exactly the same positional information, and so yeah, you're going to get that kind of regeneration. And again, the idea being you can only generate a lower positional value than your own. So, and one of the molecules we know that is responsible for this is retinoic acid. And we know a few things. So we know that if you have a sort of a distal amputation, you allow the blastema to form, but then you treat it with retinoic acid, high levels of retinoic acid. It regenerates an entire limb, just as if you had done the amputation way up here, where you have these really proximal values. So we know that the retinoic acid is responsible for that proximal specification of the limb. And we also know that retinoic acid is responsible for activating expression of PROD1. So not binding to it as NAG does, but being responsible for its activation. And so therefore, with lots of retinoic acid, you're going to have lots of PROD1, which means it's going to have the cell adhesive properties of proximal cells. And so therefore, you generate this retinoic acid and PROD1 gradient, right? Like this proximal has lots of retinoic acid, distal has very little. And so you see that. And this is a definite dose-dependent effect. So if you only put about half as much of the retinoic acid, you might still get more proximal structures, but they would be further down. And if you had just a tiny bit, you might only get a little bit of extra wrist or something like that. And if you don't treat it at all, of course, you only get the structures that are missing. So retinoic acid isn't a transcription factor itself, but it activates two homeobox transcription factors. There's more of these mice genes, but these are the two, mice 1 and 2. And so they are the ones that are responsible for the actual transcriptional activation of PROD1. So if you're looking at a blastema that is a distal blastema, and you artificially increase the expression of PROD1, they will actually start migrating to the more, if this was, where the humerus is going to form, for example. As limb outgrowth is taking place, if you activate PROD1 in this distal region, they will actually start migrating back to the more proximal location where where their adhesion matches. So where the amount of prod that they're expressing matches the amount of prod that the cells around them are expressing, which is quite an interesting observation. And also, if you disrupt or overexpress PROD1, or you disrupt or overexpress the transcription factors that activate it, you get the same phenotypes. So if you have too much PROD1, the cells are more adhesive and they will actually migrate to a new position that matches that. And if you overexpress mice 1 and mice 2, the cells will do the same thing. If you inactivate PROD1 or you reduce the amount of PROD1, then the cells have less adhesion, and they might actually migrate to a more distal position. And the same happens if you downregulate the mice transcription factors. Okay, so that was a very quick run through the whole topic, and of course there's way more. If you find this interesting, there's even some additional material still in the textbook, and there's many resources online that explain this this topic. So remember, if a newt limb is amputated, there is healing, of course, first, and it's very rapid because it covers and protects that wound, and then the tissue around, underneath the epidermal cap, dedifferentiates and divides. So we have to re-enter the cell cycle, which isn't normal in a post-mitotic tissue, and they lose their differentiation. The blastema grows, and there's outgrowth of the whole limb, and the distal structures relative to the cut site are formed. Just take note of the necessity of the nervous system, which is a source of the NAG signal. Um, and then if you do all kinds of these different grafting experiments with, different stumps and blastemas and so on, what you find is that you can create situations where this two cut pieces are connected to each other with different positional values, and they will generate the missing values between them from the proximal side. The cells from the proximal side are doing all the heavy lifting there, and under simpler levels, well, all the different kinds of experiments that are shown where there's been some kind of amputation, from the cut site in general, we see only more distal structures that are going to be formed. And then just I've listed there the most important factors. Plasmin and thrombin. I was just reading some extra stuff recently. We should add the BMPs here, because BMP is really part of this whole process too. And these are serine proteases, and they actually cleave the protein or the pre-protein, whichever it's called, to make the actual ligand. So they enhance BMP signaling specifically through cleavage. You might remember, that the TGF-beta molecules are expressed as a larger protein, but then they get cleaved, and two of the cleavage products bind to each other. This is a schematic, of course, they're not triangular proteins, but then they are bound by the receptor as dimers. And in the case of this process, a BMP47 heterodimer, right? So that's important. I'm glad I didn't forget to put that in there. So these are the main signals to remember. And hopefully you found that of interest. We will see you for the last class on Friday the 8th of August. And yes, we'll see you then.